Biologists study a wide range of organisms, which are in a wide range of shapes and sizes. For this topic, we will be discussing which tools are appropriate for which size of organism and structure within the organism. Because the course covers the organization of life from atoms, briefly, up to the level of the cell, we need to discuss microscopes. Microscopes are tools scientists use to study tissues, cells, and cell components. For this course, you'll only need to study the microscopes that I cover in this recorded lecture. So we're going to be talking about relative sizes of different organisms, some light microscopes, and the most common electron microscopes. Life comes in a wide variety of shapes and sizes. Let's go over this figure, which shows the sizes of some examples of what scientists study and the tools they use to visualize them. The human eye can see objects that are in the millimeter range up to the meter range. Well, before I had LASIK, I had trouble seeing in the meter range without my glasses. But, very nearsighted instructors aside, scientists can study multicellular organisms with the unaided eye. They can study large cells, such as chicken eggs and frog eggs, with the unaided eye as well. Once we get much past the millimeter range, we need to magnify the image of what we are studying. Light microscopes help us study objects in that range, going from larger to smaller, from millimeter to the micrometer range. That would include small cells, such as plant, animal, fungal, and bacterial cells. Some cell organelles, such as mitochondria and chloroplasts, are also visualized with light microscopes. From micrometer to nanometer, we need to use electron microscopes. They help us view smaller cell components such as ribosomes and large molecules such as enzymes, lipid bilayers, and polysaccharides. Electron microscopes can also help us visualize viruses. Let's talk about light microscopes. They use light bounced off of the objects to magnify the image we are viewing through the microscope. You don't need to learn about how the lenses in light microscopes bend light to magnify the image, but I do want you to know the size ranges of objects we study with light microscopes, which is, that's right, from millimeter to micrometer. I want you to also be able to recognize micrographs that have been taken with the various types of light microscopes. You'll be seeing micrographs throughout the textbook and in publication, so it's good to know what microscopes were used to take them. First, let's take a look at this image, which was taken using a bright field microscope. In a bright field microscope, light is beamed directly on an object. Because the objects we study using microscopes are clear, the light goes directly through them. As you can see from this micrograph, it's very hard to see a clear cell on a white background. So we add some color using a stain, like in this micrograph. It adds color to the cells, making them much easier to see. Color adds contrast between the cells and the bright background. By the way, this type of microscope, the bright field, is what you will be using in lab. This next micrograph was taken using a phase contrast microscope. Light is messed with so it doesn't go directly through the object, in this case cells. Shadows are cast, providing contrast. This allows us to see cells without staining them, viewing them in their natural state, so to speak. But not all stains are bad. The next micrograph uses stains that glow, or fluoresce, different colors used under UV light. In this micrograph, the stains are constructed so that they attach to specific cell structures. The blue stain, there we go, the blue stain attached to nuclear material, the orange stain attached to mitochondria, and the green stain attached to the skeleton of the cell, or cytoskeleton. With the cell color-coded in this way, we more easily visualize the structure of the cell. We see more detail than we would with one stain that attached to everything, like the stains we use with a bright field microscope. The type of microscope that we use with fluorescent stains is called, thankfully, a fluorescent microscope. Light can only take us to the micrometer range. If we want to look at objects smaller than bacteria or mitochondria, we need to use electrons rather than light. Once again, I won't make you learn why electrons can visualize smaller objects. You do need to know that electrons can be used to see objects in the micrometer to nanometer range, and you'll need to match the micrograph to the electron microscope used to take it. There are two types of electron microscopes. This first image was created using a scanning electron microscope, or SEM. Beams of electrons are played over the surface of an object. This allows us to see 3D images of the structures, such as these cilia that cover the surface of a human tracheal or windpipe cell. A transmission electron microscope, or TEM, beams electrons through thin sections of a specimen. That way we can look at the internal structure of the cilia. We can also look at objects that are so small that the beams of electrons pass through the object without having to slice it, such as ribosomes and viruses.